Thank you. Thank you, Zora. Thanks for the opportunity to come and speak to your group today. Welcome to everybody who's here. Really excited about this. I relish these opportunities, and I especially am interested in your thoughts and questions, and I would appreciate if you would jump in at any time. You can raise your hand here in the Zoom, and feel free to interrupt, because I'm going to be using I'm going to be trying my best not to use too much specialized language. I'm most used to talking to my neuroscience colleagues where we have a language of our own, but I'm really trying not to use that today. But I'd appreciate it if you would interrupt when you have a question, or put it in the chat, and if somebody's monitoring that, you could interrupt me, and I'd like to take the questions as I go, although hopefully we'll have some time at the end for more discussion. So as you just heard, let's see, sure. I'm the director of the medical discovery team at the University of Minnesota, and we have around 60 faculty and over 200 staff, postdocs, and students who are involved in this effort. This is just a small group of us, actually, and our mission statement really is, or our founding principle, I should say, is that addiction, substance use disorder is a biological failing, not a moral failing. And I think, it seems kind of maybe so obvious, but it's obvious to many of us in the room, not as obvious to those out in the world. And we run into people every day who do not share our view on this. And it's our goal to try to understand the biological failings that substance use disorder might be taking advantage of, and to use that biology information to design new biology-based treatment options that we believe will transform the landscape for therapy for substance use disorder, not to make a 10% difference, but to make a very large difference by providing people like yourselves, people working on the front lines, to have new tools in the toolbox for treating people who are often suffering gravely. So another part of our mission is to serve as a resource to people across the state of Minnesota. We're a state-funded institution, a state-funded organization, to provide help with community-based research projects, to serve as a resource for those, and also as a resource for educational initiatives. And so if you have needs and interests in these realms, we'd like to hear more about them. So with that, I'm going to jump into Neuroscience 101. Now, many of you have encountered this information before, no doubt. So some of it may be review, but to some of you, it may be totally new. And so I'm going to start it at the very simplest way that I can describe what the brain is doing: extracting sensory information from our surroundings and processing that information with the goal of providing appropriate behaviors as an output. Now, that it doesn't always work that way, right? Especially if some of the inputs are drugs, that those appropriate behaviors might be directed towards drug seeking and drug taking behaviors, which are really not, we don't think of as being adaptive and good for survival necessarily, right? So this is, Neuroscience of Addiction 101, not very helpful in and of itself, but a good starting point in trying, the way we think about the problem is, I don't know, can you see my pointer, by the way? Okay. So, whoops. What is happening in here? What is happening in this incredible 3-pound structure that we all have in our skulls? that can change a person's behavior to lead to these drug-seeking and drug-taking behaviors at the expense of so many other important things in their lives. I'm going to explain a little bit about how we know what's happening here. There are two principles of neuroscience that I'm going to lean on here. So bear with me as I describe these two. First one is localization of function. And I'll get to that on the next slide. The second one is neuroplasticity, the ability of the brain to change over time, to change with experience, to adapt. It's something that I hope it will happen to you today during this presentation. Your brain is going to change, and then it's going to store the information that you're learning today, and that some of that information might last a lifetime. How does that happen? What's happening in the brain that allows it to store this information for a lifetime? We still don't know the answers to that, but we have a lot of important clues based on experiments done over the years. Some of those I'll describe. So localization of function, you've probably all seen a brain diagram like this, obviously colorized here to show that there are different components, different regions that are responsible for different cognitive functions. So this is, these are always imperfect maps, but over many decades and many years of work in both human subjects and animal research subjects, where we've learned a lot of information about these different aspects of neuroscience. There are different regions that play different roles. Now, what about in addiction? What are some of the regions that we've come to understand are important? And there's a set of interconnected regions that we call the reward circuit. And this reward circuit, again, we've learned most of this information from animal models that have very similar brain layouts. Even in rats and mice, you see these similar regions that play similar functions as they do in the human brain. They're laid out in the same orientation. They have the same connections to one another. And here you see some of those regions and connections. I don't mean to attach any significance for you at this point in the names of the different regions, so they're just abbreviated here, as we scientists often do. So I'm going to describe just briefly how we came to understand what these regions do, and I'm going to lean on the animal experimentation for this, and this goes way back to the 1950s, where Olds and Milner, some of you may recall from your Psychology 101 courses where you heard about these experiments. And there's a quote I like to read here from Olds and Milner, who were doing these experiments long ago, as they described them in Scientific American back in the 50s. In the test experiment we were using, the animal was placed in a large box with corners labeled A, B, C, and D. Whenever the animal went to corner A, its brain was given a mild electric shock by the experimenter. When the test was performed on the animal with the electrode in the rhinencephalic nerve, it kept returning to corner A. After several such returns on the first day, it finally went to a different place and fell asleep. The next day, however, it seemed even more interested in corner A. At this point, we assumed the stimulus must provoke curiosity. On the second day, we did not yet think of it as a reward. Further experimentation on the same animal soon indicated, to our surprise, that the response to the stimulus was more than curiosity. On the second day, after the animal had acquired the habit of returning to corner A to be stimulated, we began trying to draw it away to corner B, giving it an electric shock whenever it took a step in that direction. Within a matter of 5 minutes, the animal was in corner B. After this, the animal could be directed to almost any spot in the box at the will of the experimenter. Every step in the right direction was paid with a small shock. On arrival at the appointed place, the animal received a longer series of shocks. That's from Scientific American in 1956 from James Olds. So for a long time, we've known that there's a system in the brain that can control behavior. And so Olds and his colleagues mapped this circuit out by placing the electrode in different places around the brain to see what sorts of behaviors or what sorts of regions could support this behavior. And this is a figure from one of their early papers showing that the animals would stimulate themselves through that electrode by pressing a lever in their cage, which they ordinarily had no interest in. But when it was connected to the small electric shocks they could deliver to themselves, then they were very interested and they would press the bar hundreds of times over the course of hours, over the course of days. So this was the beginnings of our understanding of this brain circuit that is involved in reward. And when we talk about reward, we're referring to something that the animals or people would return to again and again and get something out of. So, natural rewards would include food, opportunities for sex, building social networks. And this circuit's very important for those natural rewards. But it's also the circuit that drugs act on, and for some people that has disastrous consequences. So here's an example of in an animal models. I just wanted to show you kind of what this looks like from our lab from several years ago. Now, I'll start the video in a second here, but this is a mouse. So we got a top view here of this chamber. It's just a square box that has different cues in the different corners. And one of these corners is paired with neural stimulation. And this is actually light stimulation using a somewhat advanced version of what Olds and Milner were doing in their very early experiments, activating a very specific portion of the brain in a way that people in the past had no ability to do. But with modern technology, we can refine these things. So we call this optogenetic, that is based on light self-stimulation. Oops. Let's get this going here. So the animals, this is the first time this mouse has been in here. And you can see that one of these corners, entry into one of these corners is paired with neurostimulation. You can kind of see there's a green light that comes on the mouse's head. And that's when the light is delivered down through this optical fiber deep into the brain, into the reward circuit. So although it has the ability to traverse all over this this chamber, spends a lot of time in that upper right corner because that's the only one that's paired with this kind of neural stimulation. So this is just an example of how, Olds and Milner were right. There are circuits in the brain that will produce this do it again kind of behavior in a way that, as we've come to learn, is akin to what can happen with some people on drugs. So this is now a diagram of the rodent brain, looking at that reward circuit again. It's got all kinds of information on it, but I'm just going to point out a couple important things for our purposes today. So the first one is that, although it may not be that apparent to you, this has some striking similarities to the human diagram that I showed a minute ago. Again, and just in terms of the brain regions that are involved, how they're interconnected with one another, and something that's not present in the diagrams, what we now know, the neurochemicals are the same between humans and rodents. And so we can study the neurochemical pathways and learn about them from the animal models, and we have hope of applying that information to people. And one of the key neurochemicals here, which you've all heard of, no doubt, is dopamine. Dopamine is produced in neurons in the midbrain, in what we call the ventral tegmental area. And that dopamine, upon the appropriate signal, is released throughout different regions of the brain. But one important area is here in the forebrain, the nucleus accumbens, which in the rodent brain is actually quite large in comparison to the rest. In the human brain, it's much smaller, but retains its very strong effect on behavior and cognition in humans as it does in animals. And that's what I'll be talking about here for the next few minutes. Dopamine, we think of, well, you've often heard probably this idea that dopamine is pleasure. It's a pleasure molecule. It's released when you have pleasurable experiences. You'll see it in the popular press. This probably the most important thing I'm going to say all day is that we've learned that that is not the case. Okay, so now is it related to reward? Absolutely it is. Dopamine release is related to reward. It's related to reward in the sense that we think of this neurochemical now as neuroscientists, and there's really no disagreement in the neuroscience world about this, as a do it again chemical. It's not a pleasure molecule. When it's released, it's important for what's happening, understanding what's happening right now. The stimuli around me now are important for my survival. And if I come across these again, I need to take action to make this happen again and again, because this is important for survival. And that distinction between pleasure and survival is something that I'd really like you to take out of this presentation, because I think it reframes our view of what's happening in substance use disorder in ways that I'll describe a little bit more in a moment here. So let's take a look at some data from a rodent addiction model looking at dopamine directly. Okay, these data come from a study now quite a few years ago, a classic study that is comparing the amount of dopamine released, which is measured by a tiny a chemical probe deep into the brain of rats in that nucleus accumbens area I mentioned a minute ago. And it's monitoring the amount of dopamine released over time. And normally there's a bit released, but when nothing special is happening, it's a kind of a stable low-level signal. Now, when something happens that could be important for survival, something that needs to be recalled and perhaps done again, like there's arrival of an unexpected food source to a hungry rat, which is what happened here, there's a burst in dopamine release. Okay, and that's just something that's been replicated over and over again. Now look what happens if a rat under those conditions receives an injection of a modest dose of amphetamine. Massive dopamine signal, 10 times larger than what you see with a natural reward of a food pellet. And that, if we think about this in the context of dopamine isn't pleasure per se, but it's a signal to the brain, do this again. This could be important for your survival. Think about how much stronger this signal is here than it is for this natural reward. And thinking about it, addiction in those terms puts a different frame on it. in that, are people who are suffering with substance use disorder, are they pleasure seekers? You know, and even are they trying to avoid other things by changing their frame of mind? Maybe, but maybe there is a deep-seated unconscious signal that happens in some people that says, you got to do this again, because this is important for your survival. It may not occur to them that way because this is an unconscious signal, but nonetheless, there's a strong driver that comes from evolutionarily very old brain circuits that's saying, go get it, go get it. And that's very different than I think a lot of people's typical view of addiction and what people are going through. I've been sort of charging ahead here like a bull in a china shop. It occurs to me maybe to take a quick pause to see if there are any questions that people have.
And if there are any questions, you can feel free to unmute too. That's easier.
Sure. Yes. Okay, maybe we'll get to some at the end here. I hope there are not any questions because I'm being so unclear. There are two possibilities, right? Either everyone's getting it or nobody's getting it. Hopefully it's the former. Okay. Darn it, what's happening here? Sorry, my slides keep getting away from me here. All right. So, there's often this question, is substance use disorder a brain disease? Now, we're pretty inclined to think of it as a brain disease, and often the term kind of takes people by surprise, and maybe being diagnosed with a disease makes some feel like they're disempowered and that they can't take action and improve their circumstances. And so, we never want it to mean that. But I think it's a reasonable question to ask. Is it a brain disease? And some might say, let's look at the brain and show me where it's different. Show me the hole in the brain that tells me that addiction is a brain disease. Well, we don't see a hole in the brain. When we look at structural brain scans, and this is a bit of a silly slide here, because if you look closely, the brains on the left and the right are actually the same image. But my point is that if you took a whole slew of, healthy human brain scans and a whole slew of those suffering with substance use disorder, there's not something you can point to as a structural difference, and it's just compare that to Alzheimer's disease, compare it to, other neurological and psychiatric diseases that we talk about and study. And in certain cases, you can absolutely see a difference, but in addiction, it's not to say there couldn't be one in some cases, but in terms of like a diagnostic or something like that, there's nothing we can point to. So I like to think of it in terms of this analogy that if I don't know if any of you have been so misfortunate to have a computer virus, but if you opened up your computer to look for the virus, you're not going to find it. But if you turn on your computer, you're going to have a problem. And so, how is it that is your computer having a computer problem? Yes, it is. It's having a viral disease of a certain kind, right? But you can't point to it. It's down in the circuits. And that's how we think about what's happening in addiction. This is not a neurodegenerative disorder. It's a disorder of plasticity, a disorder of neural wiring. That wiring is changing in a fine-grained way, but a way that makes a big difference in terms of cognition and behavior for those suffering with it. And so how do we use the tools we have available to study it? That's what I'm going to spend the next portion of this presentation talking about. So here is an MRI, magnetic resonance image of a human brain, living person. It's a structural image, and it's been colorized just to show you that portion of the reward circuit that we call the nucleus accumbens, that receiving region for the dopamine that's released. So that colorized portion there indicates the position of this important small nucleus in the brain. And that's what my lab is. I'm just going to tell you kind of a short story about something going on in my lab as an example of what's happening in our team. And my lab's been studying this region for a long time. We've been studying it in rat and mouse models. And so back to this diagram for a moment, just to point out here, this is the forebrain region, the nucleus accumbens that's receiving that dopamine. So we wanted to know, how the circuitry might be changing in this area. And this circuitry, we use these computer analogies for the brain because that's the best analogies that we have to date. And we can think of these pathways from one region or the other as wires connecting circuits, and that's how we talk about them too. So are there changes in the wiring in between these different other regions and the nucleus accumbens? And that's what we sought to determine in this series of experiments. So we took a group of rats in this case, and we gave them a morphine injection each day. Not very much drug, honestly, modest doses, once per day. The rats weren't giving it to themselves in this case. We were administering it so that we could give each subject the same dosage over days and have that sort of uniformity between the subjects. And what we were really interested in was the long-term changes. And so we gave this opioid a number of times over the course of the 1 week, and then waited for a couple weeks. So the animals are in their home cages doing their thing, nothing special, and waited for a couple weeks to look at brain function. And we're very lucky here at the University of Minnesota to have an amazing resource in our Center for Magnetic Resonance Research, which is known around the world for its expertise in measuring brain function in living subjects, including mostly people. But you can imagine how much smaller a rodent brain is than a human brain. A rodent brain is like the size of the top of your pinky finger at its largest. And a human brain, you know how big that is, generally speaking. So it's tiny, and so we need an incredibly strong instrument in order to look at this tiny brain. But we do have that here, and we took advantage of it in this experiment. So I'll show you those data next. So the colorized portions of this image are showing the activity between different regions. So it's a kind of fancy mathematical way of taking an image and learning how different connections are strong or weak between different areas of this reward circuit. So where you see, we were using the nucleus accumbens as our starting place. Okay, that's what's in red here. And because the nucleus accumbens is very strongly connected to itself, it's in red, which means it's very, that's the strongest connection you can get. Now, in our control subjects, there's a small connection to the prefrontal cortex, which is always there. And that's something we expected to see. What we were unprepared to see is that after repeated opioid treatment, that there was a stronger connection to the prefrontal cortex. Now, it's just sort of, even for us, we were surprised to see that, in that we think of, okay, chronic drug exposure, that's going to, decrease different connections in the brain and make them, maybe not work as well. Well, what we're seeing in this case is that connection is stronger. It works better. That doesn't mean cognition is better. We can't draw that conclusion. But there's this change in the brain. We don't yet know what it means, but we wanted to probe how it was related to the animal's behavior, and that's in the next part of this experiment. So how do you measure an animal's behavior in relation to drug exposure? And one of the simplest ways that we and many others in our field have done it is to use this procedure that we call conditioned place preference, and sometimes we call it the opium den experiment. So the animals have this two-chambered apparatus, and they can traverse between the two different sides that have different sensory cues. In this case, we have different flooring for different tactile cues, different visual cues, so the animals can tell the left side from the right side. So on one side, they're paired with a saline injection, which is just a control, neutral kind of injection. In the other side, they get morphine. So that's done in this sort of they have an initial test where they get to wander back and forth, and neither of the chambers is paired with anything, so it's sort of neutral, like they don't really care which side they're on for the most part. They're just wandering around exploring their environment. But after being confined to one side and then the other, and having saline on one side, drug on the other, they come to associate that drug experience with we could call the opium den, right? So now they're given the choice later on. Where do you want to spend your time? Initially, it was neutral. After the drug pairing, inevitably, they spend more time on that drug paired side as a memory of a positive memory of that drug experience they want to have again. That's how we interpret these data. So that we see this increase in time spent on the drug paired side. So that's just kind of, is this addiction? No. I mean, it's a very simplified test, right? But it's been helpful in us trying to understand what are the neural underpinnings of this kind of development of a drug preference. Okay. So how do we use that in this experiment? Well, this is a sort of variant of that. Okay, so now we're getting a little bit more sophisticated with our behavioral test. This is what you've seen so far, this initial conditioning. What we're really interested in those long-term changes in the brain that might play a role in relapse. We all know that relapses, or whatever term we'd like to use for it now, return to problematic drug use, that's a really important problem that people have. You can get into recovery, you can be abstinent or be using drugs in a more less harmful way. But there are stimuli in the environment. There's stressful experiences. There's re-exposure to drug that can all provoke that sort of resumption of problematic drug use. So we want to model that in the lab. And one way to model it is after the animals have develop this conditioned place preference, if we put them back into that environment over and over again without any drug, they lose their expressed preference for that drug-paired side. So now this initial preference diminishes down to near nothing. We can provoke what we call reinstatement of this conditioned response by giving drug again. So not even drug in that environment, but drug anywhere. So we give the animals another shot of drug. They're in their home cage at that point, but then we put them in the environment and let them traverse back and forth. They're back in the opium den again, which is a pretty, gosh, sorry about this. I keep advancing when I don't mean to. A pretty consistent response that we see in our animal subjects, that when after this period of abstinence, they're given drug again, they show this, relapse-like response in our admittedly simple animal model here in the lab. So, okay, I've gone through this sort of behavioral procedure. What we were really interested in was to test what is the relationship of the brain change we identified with using the fMRI procedure with this kind of change in behavior? Are they related? If they're related, maybe we can use that information to leverage for some new possible biology-based treatment. Okay, so I'm going to the next slide here, which I'm skipping a very large number of steps, which I'm happy to discuss with you. How is it possible that we could take the opioid-treated brain and return it in this one very specific aspect to the control brain? In other words, going back here, how can we take a brain like this and turn it into a brain like this? Luckily, being here at the University of Minnesota among all sorts of people who have technical expertise that is way beyond me and way beyond my colleague Ben, who's here with me today, we were able to engineer a way to do that quite specifically, to take this connection, which was strengthened after opioids, and change it back to this kind of a state. Okay, and that's what is in the slide that I'm showing here, with some details about how we did that and how we measured that brain state was returned to normal. And I can go into some of those details if you're interested, but it would take me quite a long time to explain all of them here. So I'm just cutting to the chase, which is we used this procedure after the mice had been treated with opioids, had expressed their preference for the opium den, and then went through an abstinence period. We gave them neurostimulation at that point, which involved neurostimulation with the light pulses, like you saw in that the chamber where the mice were seeking the light pulses. We gave them a pattern of light pulses which produced plasticity, returning the brain to a normal state, and then we gave them the re-exposure to opioids that would normally provoke a relapse response. This is what we saw in behavior, okay? So all the different control groups are on here, so it's a little bit more, involved than I can go through that in detail if you'd like. But we see this preference develop with our standard model. We see that with abstinence and extinction, we see that go away. With drug reintroduction, this is our normal kind of relapse-like response that we see with the opioid re-exposure. But this is those animals after they've received the neurostimulation that returned the brain more to a normal state. And what we find is that even though they've been exposed to that additional dose of opioids, they don't show the relapse-like response that the other animals do. And that is, you know, in some ways, it's a small animal experiment in the lab with a, you know, a very simplified model of addiction. But what it gives to us is hope that we can use this kind of information about undoing some of that rewiring in the brain in order to, you know, to help people who are, you know, have been suffering with their own problematic drug use, are now in abstinence, but need another tool in the toolbox to prevent against relapse, which, as we know, especially for opioids, can be fatal. So you know, what do we do with this kind of information to get it to be of use to anybody out in the world? Well, that's in part why we have this large team of researchers who are working at all different levels to try to take information, to first of all, to generate information like this using our animal models in the lab and using the best technology that we have available to study what's happening in the brain. But then to take that information and to translate it into possible human usage. And we have some amazing people. We have some amazing resources in terms of the tools and technology we have available here. We have an amazing community of people in Minnesota who are very invested in you know, trying to address substance use disorder from all different angles, you all included. And so we feel very hopeful that we can, by working together, come to some really new ways to deal with what is a very problematic issue in our society. So how do we, you know, I mentioned we have this way of stimulating the mouse brain with light in order to get very specific kind of stimulation to produce plasticity that might be beneficial. How do we do that in people? You know, I mean, there are uses for surgery, surgical methods to implant electrodes down into the brain. Maybe some of you have heard of deep brain stimulation. It's often used now in Parkinson's disease for people who have drug intractable symptoms of Parkinson's disease that are debilitating for them. That's been in use for decades now. You know, I think that's a possible avenue, and some are already taking that avenue. There are studies in China. There's some studies now in Virginia and West Virginia here where opioid patients are being implanted with electrodes for stimulation. Some of you might have seen the recent 60 Minutes episode. But we're hopeful that you know, that surgery won't be our final option. And on that front, there are a number of different means that we have available now for what we call non-invasive neurostimulation, using things like transcranial magnetic stimulation and transcranial direct current stimulation across the scalp. So using, you know, there's no surgery needed here. We have some experts in the world who are here at the University of Minnesota, know how to use these techniques, know how to keep advancing the research behind them so that we can, you know, not only use them in their current form, but continue to improve them. And that's the sort of way that we can take the experiment or the data coming out of the mouse experiments and begin to understand, you know, how we might modify the circuitry in people in a beneficial way, using these non-invasive neurostimulation approaches. Now, this isn't something like electroconvulsive shock therapy, right? I mean, it's related to it in that it's electrical information being delivered to the skull and ultimately to the brain, but we're not producing seizures here. We're not, you know, these are mild stimuli that people in many cases can barely detect, but they're having a specific effect on specific neural targets. And our job is to figure out what are those targets and how do we engage them in a beneficial way. And that's a big part of where our team is headed. Using devices that, might even be available for use at home and in treatment facilities. And so not even necessarily something you'd have to have a doctor's visit for. So this is really on the frontier, and we're not there yet, but we're getting closer and closer to the point where we could actually implement some of these things. Thank you for your attention. I think we have 10 minutes left for any questions and comments. As I said at the beginning, I'm very interested to hear your thoughts and your questions. So fire away. Thank you.
Thank you so much, Dr. Mark. That was super interesting. Do we have any questions? Feel free to write them in the chat or unmute.
I have a question. This is Dana Hayes. I work at Wayside Recovery Center. I am going back to the beginning of what you were talking about and that popular idea that it's so easy to connect to of the dopamine as a pleasure drug. So it feels good. So I want more of that.
Yeah.
Or which it's easy to a unscientific mind to think, to kind of connect that with evolution too, like that graph about the food, like I need food to survive. So I'll get some more food somehow, or I'll devote myself to that.
Yeah.
But how I'm just intrigued by it sounds like, or is this even, I may have just misinterpreted, that part of what you've found is that it's more a stimulation than a pleasure, a stimulation that isn't necessarily the same as pleasure.
Yeah.
Is that, I don't know, can you just speak about that a little bit? It's a little, it's less intuitive.
Yeah, it is less intuitive, and many of us studying this in detail have really struggled with it for many years, and that's why the message, I think, hasn't gotten out and isn't more widely understood, because it's a little more murky than dopamine equals pleasure. But this idea of do it again, I think is a good way to describe it. In this particular circuit, the meaning of dopamine seems to be whatever happened just now is significant, do it again. And I didn't show you the experimental data for that, because actually it took a long time and a lot of different experiments, mostly in the animal realm, to be able to determine that dopamine wasn't signaling the pleasure itself. It was signaling something that usually goes along with the pleasure. Because the idea is, from a biological perspective, they co-evolve together, that the pleasure, which we really think is the endogenous opioids or the endorphins, as you've heard, that's the other term for endogenous opioids. That's the system that really is the pleasure system. And it usually works in tandem with the dopamine system, but not always. And that finding, it's a little bit of a sort of, seems like a nuanced finding, but to us, it's like a really important one for the message of substance use disorder in particular, because it's, to me anyway, what it means is it's not people chasing that euphoria, and they're not the pleasure seekers. There's a deeper signal that's harder even, for any individual to understand. Why am I doing this? I don't want to be doing this. But yet they're drawn to it. Why are they drawn to it? We think it's because this do it again signal is ramped up to such a degree that it's hard to battle it, you know? I don't know if that helps answer your question, but hopefully. Thanks, Dana. Oh, here, I see one from Andrea. Can medication produce plasticity? What is the research saying in the success of TMS? Yeah, this is a great question that that we get a lot and we don't have a good answer. I mean, absolutely, for different kinds of psychiatric medications, they do produce plasticity. Not all of it is beneficial, but I mean, like, you know, there is some of it that certainly is. But we don't really have as much understanding of the exact plasticity that medications are producing that we'd like to have. I mean, this goes for depression drugs, for anxiety drugs, for basically drugs for any psychiatric condition. It's a harder question to answer than we thought it would be. But I mean, we're talking about medications like methadone, buprenorphine. I mean, those are clearly producing beneficial effects. They're probably not producing as much beneficial plasticity as we'd like them to be. And the evidence for that is that if people stop taking them, it doesn't seem, you know, there's no lasting effect of the drugs themselves. Maybe people have used them to get a better footing, to get more therapy and psychosocial therapies to the point now where they are stabilized, and that, you know, that's fantastic. And there's probably neuroplasticity that relates to that, but we don't know the answer to that. In fact, I recently got asked to speak to the Yale Psychiatry Department, and one of the people who was there was saying, like, you and I got to study that. And so, you know, we haven't formed a plan for it yet, but to me, it's a really important thing that we should be studying, because if we can learn about what that plasticity is, maybe we can find other inroads to use these, you know, newfangled approaches to make it even more powerful, like TMS, which is part of the question here. Thanks, Andrea. Damon asks about mindfulness exercises to produce plasticity, and we, again, our very strong suspicion is that there is beneficial plasticity produced by those mindfulness exercises, and there are very few people who have the hard data on that, but it's beginning. People are very interested in it, and it's beginning to be studied in more mechanistic and detailed ways in terms of the biology. But as I often mention to my students in sort of a joking way, but like, we're going to study all this stuff and then we're going to figure out that we had the answer, you know, 2,000 years ago. Just meditation and, you know, I think there's a real possibility that combined approaches are going to be, you know, what really does the job. You know, we've got in some cases medications combined with psychosocial treatments, maybe a dose of neurostimulation at the precise time. You know, that's to me where the future is combining these things, and that would include mindfulness and yoga, journaling, you know, things that are other ways to produce plasticity than having electrical stimulation across the scalp. Well, maybe time for one more.
Any last
Thanks, Margot. It's my pleasure to be with you. I have a real passion for the subject and for getting this information out there. And so if any of you have other opportunities and would like a talk from me or from my colleague Ben, you know, we'd be happy to. And as I mentioned, community-based research projects. We're very interested in those opportunities as well. We want to be a resource for people. So thanks for the opportunity to speak with you all.
Yeah, thank you so much. Thank you, Dr. Mark, and thank you everyone who joined today. Just a little bit of follow-up information. If you need a certificate for CEU for this, please just look out for a survey that I will be sending out in the next couple of days. Fill that out. You should be able to indicate if you need CEU on that, and then give me a couple weeks and I'll get those certificates out to everyone. So. Thank you again for joining and fabulous presentation. Thank you so much, Dr.
Thanks. Take care.
Thank you.